Hi everyone. In this lab, I'm going to talk about the effect of osmotic pressure. We're basically going to discuss what happens when you take different bacterial species and you grow them on agar plates with different salt concentrations. And the point of this lab is to see how salt or different solutes affect cells. And this is relevant because it helps us see when we're growing bacteria in lab what salt concentration to put in the agar or broth. It also helps medically because if we're trying to get rid of bacteria, we know what salt concentrations can potentially kill them. So we're going to talk about osmotic pressure, and then we will talk about the experiment. To give some background, water is very critical for all cells. So just like it's critical for our cells, it's critical for bacterial cells for them to do their metabolism and grow. The definition of osmosis is the movement of water across a membrane, so a cell membrane. And remember, osmosis is a form of passive diffusion. So water can just go in and out of cells with no energy. And keep that in mind. A lot of students confuse it and they think that water is just going into cells, but just like water is going into cells, it's also leaving cells at different rates. And that's the definition of osmosis. Again, it's just the passive diffusion of water across cells. So the direction of the water, whether it's going more in the cell or more outside of the cell, that's determined by the amount of salt or solutes. And solutes can be sugars, can be different salts. So the water will move where there is always higher salt concentration. So water moves where there's less of it, and there's less of it when there's a high salt concentration. So keep that in mind. So if you have a cell and it's in solution, if there's more salt inside the cell, water will move in the cell. If there is more salt outside the cell, water will move outside the cell. So water will always move where there's more salt. And that's what osmotic pressure refers to when we talk about this. It's basically saying, well, how will water flow when we have different solute concentration? That's what osmotic pressure is. And osmotic pressure is important for all cells. So it's important for bacterial cells. It's important for our cells. That's why when someone is dehydrated, we give them an IV in the hospital. You don't just give that, an IV is not just plain water. It has different solute concentrations, and there's different solute concentrations so that our cells stay healthy. So the different types of solutions that we can put cells in, there's three types of solutions in science. There's isotonic solution, hypotonic solution, and hypertonic solution. And I want everyone to keep in mind that when we use these terms, we're referring to the actual solution, not the cell. I'm not saying a hypotonic cell, I'm saying a hypotonic solution. So all of these terms refer to what are we putting the cell in. An isotonic solution, if you put your cells, whether it's your red blood cells or bacterial cells, in an isotonic solution means that the solution has equal concentration of salts as the cells. So water moves in and out of cells at an equal rate. That's the equivalent of an IV. An IV, typically when we put it in humans, it typically is isotonic to our cells. So it has very similar salt concentrations to our cells. Therefore, the movement of water in and out of cells is equal. So we say the net movement of water is zero. That doesn't mean that water is not moving. Water is always moving in and out of cells. It just means that it's going at an equal rate. So whenever you put a cell in an isotonic solution, water is going in and out at an equal rate. If we take a cell and we put it in a hypotonic solution, the word hypotonic means that the solution has less salts than the cell, meaning the cell has more salts than the outside solution. And remember, water will always go where there's more salts. So when we have a cell in a hypotonic solution, water will enter the cells because the cells have more salt than the hypotonic solution. And when you have so much water entering the cell will get bigger, and it could potentially burst. So we say when we put cells in hypotonic solution, we get osmotic lysis. So the cell bursts because so much water is going in. This is why it's bad to overwater plants. You can kill plant cells just like you can with bacteria. Hypertonic solution is a solution that has more salts than the cell. So hyper means more, and so a hypertonic solution has more salts than the cell. So then then the water in the cell will go outside the cell, because again, the water will always go where there's more salt. And when water keeps leaving the cell will get smaller and smaller and smaller and shrivel or shrink, and we call that plasmolysis. So that's the result of a hypertonic solution. So just take time going over these, and please don't memorize them. Think them through, and you can think your way through any exam once these make sense. Always remember that with osmosis is the passive diffusion of water across cell membranes, and water always goes where there's a higher salt concentration. So if there's a higher salt concentration on the outside of the cell, that's a hypertonic solution. Water will leave the cell and the cell will shrink and shrivel, which is bad for the cell. If there is a higher salt concentration in the cell than the solution, that's a hypotonic solution. Water will enter the cell and the cells will just burst the lyse. So that's also not a great solution to put cells in if we are trying to grow cells. An isotonic solution is the best solution to put cells in because it has equal salt concentration as a cell. So water will go in and out of cells at the same rate. So that's why we say the net movement of water is 0. Okay, so now that you guys know this, most microbes cannot tolerate hypertonic solutions. And the reason they cannot is, remember, if we take a cell and we put it in a hypertonic solution, hypertonic, hyper means more. A hypertonic has more salt than the cell. Therefore, the water will leave the cell. So when we put cells in a hypertonic solution, the cells will shrivel because water will keep leaving them. And this is not a good thing. So most cells will die when we grow them at high salt concentrations. That's why we salt foods. That's essentially the reason why really salty beef jerky can last on the shelf for many days, probably years. So salty food lasts a very long time because it doesn't really grow bacteria, because if the bacteria were to try to grow there, their cells would lose all their water, they would shrivel. So that's what happens in cells. Now, some cells, few, have evolved mechanisms to live in high salt or high sugar environments, and we call these halophiles. Halo means salt. Phile is the opposite of phobia. So a halophile is an organism that loves salt. So it's a salt-loving organism, and they typically live in salt conditions that are much higher than the average salt conditions. So the average salt conditions that most organisms like is between 0% no salt to 1% salt, and halophiles like salt that's much higher than that. The reason that they can survive in these environments, because I just listed all these things about why most organisms do not like salty conditions. Well, these cells, halophiles, can survive because of 2 reasons. One is that they have increased internal solute concentrations. So if their solute concentration is really high and you put them in a salty environment, it doesn't make a difference because their internal solute concentration is high, so it doesn't really change the movement of water. And also a lot of halophiles have pumps out of this, they have different proteins on their cell membranes that pump salt out of the cell. So it's beneficial for them to be in salty environments because of these 2 reasons. Now, there's 2 groups of halophiles I want everyone to know. There's what we call facultative halophiles. They mean the same thing as halotolerant organisms. And then there's obligate halophiles. A facultative halophile, which is the same as a halotolerant organism, means that these organisms can tolerate salt. They're not killed by it. So they can live in normal to moderately salty conditions where most organisms cannot. So if an organism can live in up to like 10 or 15% and sodium chloride, which is salt concentrations, we call it a facultative halophile or halotolerant organism. So it can tolerate salt. An obligate halophile, obligate means you need, and halophile is salt-loving, means these organisms will only grow, they're obligated to grow in high salt concentration environments. If there's not high salt, they won't grow, they will die. So they need high salt to grow. They will only grow in high salt. That's why we call them obligate halophiles, which is the same thing as extreme halophiles. They require at least 9% sodium chloride, like about 10% salt conditions to actually grow. And examples of obligate halophiles are organisms of the domain Archaea. Remember, Archaea are not bacteria. bacteria. Bacteria and archaea are both prokaryotes. They're both microbes. Archaea are just organisms that live in extreme environments typically. So there's a lot of archaea organisms that are obligate halophiles. They only grow in super salty like lakes or brine. And then an example of a bacterium that is an obligate halophile is Halobacterium salinarium. It grows typically in salt lakes. I didn't mention this, but an example of a facultative halophile, an organism that tolerates high salt, is Staphylococcus epidermis, which is bacteria that's typically found on our skin. And remember, our skin deals with so many different salt concentrations. So here's an image of Staphylococcus epidermis, again, which is a facultative halophile, and here's an image of Halobacterium salinarium, which is an obligate halophile. Now, for this lab, the goal of this lab is for you guys to take 4 different bacterial species and determine what salt concentrations they need to grow. So what you were supposed to do in lab, if you were actually doing it, is we had different agar plates, so 5 different agar plates. An agar plate with 0% salt, an agar plate with 1% salt, and when I say salt, I'm referring to sodium chloride, an agar plate with 10% sodium chloride, 15% sodium chloride, 25% sodium chloride. So 0, 1, 10, 15, 25% salt, and you were supposed to streak different bacteria on all of these agar plates, and you were supposed to see where do they grow. So the bacteria that you were going to streak were Bacillus subtilis, which is a typical soil bacteria, E. coli, which is found everywhere, Halobacterium salinarium, which is found in salt lakes, and Staphylococcus epidermis, which is a skin bacterium. So you are going to streak them on the agar plates, incubate the plates, and look at the results. This is the results that you should have seen. So we're going to go through all the plates and go through all the bacteria. So right here, this is the agar plate that has no extra salt added, so 0% sodium chloride. This is the plate with 1% sodium chloride, 10% sodium chloride, 15% sodium chloride, and 25%. So no salt to very high salt, 25%. That's the order. Okay, the first streak of bacteria is Bacillus subtilis. So when I look at Bacillus subtilis, it grows on the plate with no salt. It grows on the plate with 1% salt. It grows on the plate with 10% salt. I see no growth on 15% salt and no growth on 25% salt. So I would say for Bacillus subtilis, I would classify this organism as not a halophile because it does not really grow on these, it doesn't grow on these plates, and its growth is, it starts growing very little on the higher salt concentrations. Now we look at E. coli. E. coli grows on the plate with no salt. It grows on the plate with 1% salt. It does not grow at 10% salt, no growth at 15%, and no growth at 25%. So because it only grows at 0 and 1%, I'm going to say that this is not a halophile either. Now we look at Halobacterium salinarium, which was supposed to be streaked here. I see no growth at 0% salt, no growth at 1%, no growth at 10%, no growth at 15%, but there I see bacterial growth at 25%. So this bacteria only grew on the agar plate with a very high salt concentrations, 25% sodium chloride. Thus, I would classify Halobacterium salinarium as an obligate halophile or an extreme halophile, meaning that this bacteria absolutely needs high salt concentrations to grow. It doesn't grow on any of the other plates. Now I'm going to look at Staphylococcus epidermis. So Staphylococcus epidermis, I see growth at 0% salt. I see growth at 1% salt, growth at 10% salt, growth at 15% salt, and no growth at 25% salt. So it grows on 4 out of the 5 plates. So I would classify this as a facultative halophile. It can tolerate high salt, but obviously at the very high salt concentrations, it's killed. But it is facultative or a halotolerant organism. It can tolerate these different salt conditions. So go ahead and fill the effect of osmotic pressure table in your lab manual. And what I want you to get from this lab, so let's go over the questions. So I want everyone to know what these terms mean: hypertonic, hypotonic, and isotonic, and know that in this experiment, we were addressing high salt conditions or solutions. So we would call that hypertonic solutions. Classify the organisms in lab. So we just went through all the organisms. We looked at Bacillus subtilis, which was the first one. We looked at E. coli. We looked at Halobacterium salinarium and Staphylococcus epidermis. So based on looking at these plates, I determined that Bacillus subtilis and E. coli were not halophiles. Halobacterium salinarium, which only grew at high salt concentrations, I said that I'm going to classify that as an obligate halophile. And for Staphylococcus epidermis, which is this last streak, it grew on all of these plates except 25%, so I will classify it as a facultative halophile. Explain why Staphylococcus epidermis had the widest range of salt tolerance. So I want you all to think about this. So Staphylococcus epidermis is bacteria that's found on our skin, and think your skin deals with so many different salt conditions. So it makes sense that for this organism to thrive on our skin, it would need to be a facultative halophile, because sometimes you're sweating and that's very salty, and sometimes you're not. So it has to deal with different salt conditions. When we look at the 4 bacteria we studied, the organism that had the most growth at the highest salt concentration, the 25% agar plate, the only one that grew was Halobacterium salinarium. And so we classify that as an obligate halophile. In the beginning of this lecture, I talked about how do halophiles avoid plasmolysis in salty environments. So remember, when you put I was going to say normal bacteria, not a halophile, in a hypertonic environment, a salty environment, typically what happens is the water will leave the cell will shrivel or plasmolysis will occur. The reason that halophiles can deal with salty environments is that they either pump salt out of their cells or they have a high internal solute concentration. Does salting a fish protect it against all spoilage organisms? So I want you guys to think about this. If you salt a fish, will you protect it against all bacteria in the world? What about halophiles? So think about that. And why is it possible to keep nutrient-rich honey at room temperature and still avoid bacterial growth? Why is it that we salt foods in general? And then know that Halobacterium salinarium was an obligate halophile. So go through all of these examples and keep in mind that there are all types of bacteria, and salting does not protect against all potential spoilage organisms because some are halophiles. But in general, salting is really good because it does avoid a lot of bacterial growth because of the hypertonic environment that causes the cells to shrink or plasmolysis to occur.